दबाओगे तो आप it will be
Okay, may I please have your attention, please? Can you take your seats? We had a small extended break because of some issues on the display, and the issue was another kind of conflict which has started here because of rats, rodents. They're cutting the cables inside. Okay, so we have monkey conflict, we have now rodent conflict. So we will start with the next session, and to begin with, I will request Sri Ajay Saxena, former PCCF and half of Arunachal as well as Goa. Saxena Sahab has been a faculty member at this institute, so has a very long association with Wildlife Institute of India. Welcome, sir. I request Dr. Vaibhav Mathur, who is again a former diploma trainee of this institute and belongs to Assam cadre. Please take the chair, and Professor Kamar Kuresi to co-chair this session.
If you can call the people from outside, the ones who are at the poster session. Bipin, if you can call the people over there. I request session chair to start the session, please.
Thank you, Kamar. It's always a pleasure to come back to the institute. I've seen it growing from foundation stone when I was a probationer in Forest Service, and diploma training, and then faculty also. So it's a pleasure, and it's one of the best place to be in. We have got excellent sessions in the morning and post lunch, which is always a difficult period to listen and be attentive. And I hope after tea break, you people are more attentive and active. Session is actually focused on, again, as in the previous session, the chairman said, the charismatic species. Yes, it is also focusing on charismatic species, and starting from, it's on landscape level population dynamics of these species, mostly focusing on central India, but then it's also coming to the foothills, and then climbing up right up to the Ladakh, and that's quite interesting. And there, the species which are being described are quite different. No charisma other than snow leopard, but the main player in that study is quite interesting, and we have discussion on that. I'm looking forward to it. So, Kamar, please take over, and I will be silent.
Namaskar. I invite now Dr. Bilal. Dr. Bilal is our large predator. He replaced the old large predator. And he is expanding his territory from wolf to tiger. Now he's occupying the elephant landscape. Over to Bilal.
Good evening, sir. I never realized or never thought in my life that I'll be talking about elephants, and that too about a problem or an issue which, that too so late. This is after 16 years of, in WI. But yes, an opportunity came, and I thought, yes, let's give a try to it. And the issue is, everybody knows that we have 3 species of elephants, and there are a lot of elephants outside Asia, and Asian ones are around 40,000 to 50,000. That's what estimates say. But if you see the Indian estimates, you always have an elephant numbers which are given is something around 29,223. When you see this type of counting, it really mesmerizes. And when you talk people, what they say that, oh wow, elephants are huge, they are visible, you can count them all. And that's probably why you see that type of numbers, because that's what the perception is. And people also perceive probably that elephants gather in water holes. If you go in the summer, you will see all the elephants. And people also think that, oh, they all come together, they live in a groups, and the counting is much more easy. And people also feel, because most of us, as a childhood, we see a lot of African elephants in savannas, and you see then aerial photographs, and then probably you realize all the elephants are in open habitats, and you can see all of them. But that's not what the problem is. There are a lot of elephants which are in dense forested habitats. They have a very large ranges, and there's a very interesting group dynamics. They have a fission and fusion, and they also have aggressive behavior, and there are also a limited resources when we talk about the population estimation of the elephants. So yes, this is a very simple slide, which typically represents elephant behavior. And people probably will say that, yeah, we can count them, but there are so many elephants which are behind the elephant. And so many measures. Okay, somebody says that the ear is this, somebody says the tail hair is upside down, somebody says that no, tusk is broken. So these are all measures probably which are used to identify elephants. But that's only possible if you sustain this. Because as you see elephant in a forest, what's the first thing you will do? You will run away. And looking for those smaller measures which people have educated for monitoring elephants are doing, and elephant populations, it's very good if you are making like an atlas of elephants and identifying elephants, because population estimation is a very short duration. So you have a month or 2. So under such circumstances, what's to be done? So I have just put the estimates which we have. We have something around 29,964 elephants in our country. And we have a very precise estimates, 4,015 elephants in Tamil Nadu, 6,068 or 6,049 in Karnataka, and so on. So how we can change this particular process, or we can have a better options to estimate or have a number of elephants in our country. So we started with discussions with Project Elephant, and then they said, yes, we probably need to think about a popular, the methods, which probably we can develop into a national level protocol. So we decided that, okay, what's to be done? We said that, yes, everybody does a camera trapping, let's put camera traps. We said that, okay, sometimes people also go as a human observers do that, and the genetics, which is considered as one of the important tools. And I will give you the one by one about what type of methods we use it. So the first of all, we tested our dung decay, and we did most of this in the western part of the Rajaji Tiger Reserve, believing that it's a closed population because there is no movement of the elephants anywhere. So it's a very nice experimental site to test the methods and to visualize or to check whether your estimates, whether your methods are actually performing or not. So in December 23, we did something around 40 transects. We walked around 522 kilometers, and we tagged around 320 dung piles in Rajaji. And we estimate the dung decay rate, which was around 0.044 per day. We estimated dung density. We converted this. We used a defecation rate, which is already there for elephants. And we estimated the population. The population came around something 223, with a variation from 92 to 554. Now, if you take this estimate, it's perfectly fine. But if there is a 50 or 20%, 10% decline in elephant population, you will never know it. If the Rajaji loses 10% of the elephants, by this estimate, you will never know that whether elephant population has decreased or increased. And the CV of this particular method came to be around something 52%. And again, 1% error in decay rate gives the 1% error in population estimation. So for example, you have something around 12% variation in the decay rate, which we had in Rajaji. So imagine you will have a 12% variation in dung decay. And then dung decay varies from one place to other place. Like there are environmental variables, there are microclimate, there are abundance. And dung decay takes a lot. Some dung piles have decayed for 245 days. So you have to wait for 245 days to estimate the population of elephants. So this was this particular method. And people nowadays use a lot of camera traps. So we also divided the Rajaji in a grid of 2 square kilometers, placed camera traps at the center of the grid, which is one of the assumptions of when you are doing camera trap-based distance sampling. We have something around 9,342 trap days. And we got elephants on 51 camera traps. So when we try to estimate the population, in this particular method, you don't need to identify an individual. Okay, and we got something around density of 1.21, a CV of around 35%, and we got a population of 700 individuals, and the limits were 358 and 1,398 elephants. So imagine the variation. One method gave you something around 300 elephants. The other method is giving you around 358 to 1,398 elephants. And if there's a 50% decline in Rajaji by this method, you will never detect anything. And we again, there's one more model which a lot of people are using. It's a random encounter model. So same data which was placed in the center of the grid, random traps. The REM assumes that animals move randomly in an area, and the detection is a factor of abundance versus speed. If the animals move faster, if there's a higher abundance, probably you will have more captures. We used the same method, calculated the population, which came around 221, and 157 to 285 was the rate, and CV was something around 29%. And then we decided that, okay, we will also put camera traps not at the center of the grid. We decided to put the camera traps at the preferred routes within a grid. So the grid size was 2 square kilometer only, but the camera traps were placed at a place where there is a higher possibility of capturing the elephants. And we also changed the routine. So what we did, we placed one camera trap at the height of which we normally put for the tigers, and one camera trap which put slightly up, so that if there's an elephant coming in front of this particular camera trap, there's a better probability of looking at the elephants, identifying the elephants, or getting a full group composition. And we ran a model based on special partial identity. What we did here, we identified that only some of the elephants are identifiable, in the sense like you have a tuskers, you have some elephant which has some specific mark on the body. So we used these partial identities and then tried to estimate the population. We found that the population was around 273 with a variation from 143 to 403, and the CV was something around 24%. We also done, we thought about like, can we do something else, resource-based capture. We thought that during summer months, water is a very important resource for elephants. Okay, and if you look at the camera trapping 2x2 grid size, we were using something around 222 grids, something around 466 camera traps for doing the analysis. We found that Rajaji, this part of the Rajaji has 46, has some 46 water holes. We tried to cover during summer month, considering that the presence of a water is a critical resource for elephants during the winter, during the summer, and presuming that most of the elephants will come to the water hole for drinking the water. And we calculated we identified something around 18 groups. We estimated the population, which was around 179, and which came around, the range was around 172 to 187. So we also did a direct line transit. And I, like Vivash has sent me so many times with the diploma trainees to do the line transit for elephants. And every time when we encountered an elephant on a line transit, we return it back. Okay. Now the challenge was going on a line transit after seeing an elephant, completing that line transit. So I took some 30, 40 researchers. I said that, okay, let's do. We did a line transit. We found a number of elephants to be 122, and the range ranged from 70 to 174. But the CV of this particular method was something around 40% CV, right? And then we also took the help of Dr. S.P. Goyal, and we collected a lot of dung. Okay, and we said that we should also try to evaluate the efficacy of the dung methods for the population estimation. So number of samples around 107. We got, this is the dung which we collected on the line transit, and the population estimation was around 76, which was from 66 to 84. We did a opportunistic dung collection, like we didn't move on the line transits, we moved everywhere. We collected around 1,000 dung, elephant dung, and out of which 600 were processed, 200 used it for the analysis, and we found around 90 individuals, and the population varied around 84 to 85. And just 2 years before, a very interesting paper came in ecology, which is a special capture-recapture model for group living species. We thought, wow, all the problem is solved. Now there is a method for group living species. But what is this problem in this particular group living species? It's a probabilistic assignment of individuals to the group. What does it mean? If you have a one right side photograph, you don't have a left side photograph. So this model will consider all the left side photographs is the probability of this one right side photograph. This was the one issue with this particular model. The other important model was, this model even assumes 0 as a group size. If you don't have a capture, it assumes that. We thought this is a good model, but needs some modification. So what we did, we used the elephant ecology to increase the efficacy of that model. So what we said, elephant is a social group. The movement of elephants is governed into that particular social group. So wherever you get an elephant capture, you get a GPS location of that particular camera trap. And we also put that there will be a relationship between the group size and the number of elephants photographed. So if there is a group of 25, if it is moving in front of a camera trap, you will not get 25 elephants, but you may get 10 elephants, 12 elephants, or 13 elephants, because you have a difference in like delay in the camera trap. So we used that. Then we considered that group size as a weight of that particular camera trap, and we considered the distance, daily distance moved by an elephant. So we used the average speed, which is how far an elephant can walk in a one day, which is a 7 kilometer. And we identified the clusters first. So that's the first part. So we try to identify how many groups are there possible in a Rajaji. Then what we did, we used a maximum livelihood approach to estimate if camera trap has got a photograph of 12 individuals, how many individuals have been missed. So if you see the bottom lines, these are the equations for calculating for special clustering. We call it a special clustering, and then estimating the number of individuals. So what was interesting, we used two different methods. So one, we used the data from the camera traps, which we placed at the center of the grid, and other, we used the camera traps, which we placed at the normal trails, but all in 2 square kilometers grids. So the cluster where the camera traps were placed in the center of grid, we got 5 groups, and the population estimation was around 50 to 55, whereas in random model, we got around 128 animals, 9 groups, 128 animals, and the population size was around 124 to 131 animals. And this is about, the CV was around 5.16%, and in other, the CV was around 11.17%. We thought we should validate this, whether this clustering model is right or wrong. What we did, we actually identified some of the elephants which were identifiable. Now, if our model is correct, that means those identifiable elephants should fall within that particular cluster. So we found that we got 4 different types of groups where we got recaptures, and we thought all the elephants which probably were that group, they were actually assigned to that particular group. In the meantime, Goel Saab came up with a map of the dung piles, because dung piles were also identified in cluster-based model. And we tried to map the dung piles, and dung piles, the accumulation of the dung piles inside the Rajaji Tiger Reserve actually behaved like a cluster. So if you see that center one is a dung-based cluster, which is the actual clustering of dung piles, versus the cluster which you done because of the camera traps. So this is all the methods, the population you have and the CVs you have, and this is what. So imagine same 580 square kilometer of area, 11 different methods, 11 different results, okay? And see the variation in CV. We have a CV varying from 50, we have a CV which goes up to just 5.2. So now we are trying to evaluate. So there is an issue of the precision and the bias. Now, which model gives you the precise estimate which is unbiased? There may be the models which are biased. So we also try to estimate the cost. So which method should we go up, which will give you probably a better model? So if you consider 1% CV, 10% CV, and these models are performing better, which is like clustering SECR and the genetic-based and resource-based models. But resource-based models, they're very precise, but we don't know whether they are biased or not. I think they are far more biased. So this is what we are thinking about, working on this particular issue and trying to see which model probably will be the best fit for estimating the elephants in our country. Thank you.
I now invite Jayant. Jayant holds a master degree in environmental science. He joined All India Tiger Monitoring in 2014. He's from Assam. And as Bihu dance, he's also very quiet and subtle in his moves, in his data analysis. Over to Jayant.
Okay. Hello. Yeah. Good afternoon to all of you. I am starting by talking about my project. It is a long-term tiger monitoring project initiated by my supervisors Kumar Qureshi and YV Jhala with the support of NTCA and Madhya Pradesh Forest Department. The major objectives of the project are monitoring tiger, co-predator, prey and their habitat. So far, and also developing and testing methodologies for All India tiger estimation. So far, we have worked upon population trend and demography of tiger, leopard and their major prey, and also we have worked on some other aspects. The project outcomes have been incorporated in the tiger conservation plans, also in science-based conservation management. We also have some publications under the ambit of this project. Now coming to my topic, large carnivores are an essential component of the ecosystem. However, they are facing massive declines. Being at the top of the food web, they occur at low density and susceptible to the demographic changes. The demographic rates are influenced by these key ecological mechanisms. First is top-down control by the apex predators, bottom-up limitations from the food availability, and intrinsic regulations through the inherent density-dependent factors. Leopard is a solitary large carnivore. However, they are widely distributed from sub-Saharan Africa to the Russian Far East. However, they have now exist in only 25% of their historic range. India has lost 75 to 90% of their leopard population in the past century. Currently, we have 14,000 leopards in India in the tiger bearing forests. However, interestingly, the leopard population is not is stable, unlike the tigers, which are growing at 6% annually. The major reason for this could be the human-induced mortalities, as more than 67% of the population actually remain outside protected areas. Several reports have showed that tiger population, there are around 200 leopard poaching cases detected annually. However, this could be more than 10 times higher according to custom authorities, which should be around 14% of the population. So, let us look at how the ecological mechanism I have just mentioned applies to the leopard population dynamics. Leopards occur symmetrically along with tiger and lion across their range, and several studies have showed that tiger and lion densities impact negatively the leopard densities. But how it influences their demography, it is not known. Again, high density and the large leopard populations occur in the protected areas and tiger reserves, which also has high prey densities. So, it can be, it is likely that leopard densities are governed by prey abundance, that is the bottom up limitations. However, leopards are also occur in human dominated landscapes and subsisting on livestock and dogs. So, this raises a question how wild prey availability impacts their demography. Since leopards are territorial species, they are also subjected to interspecific competition. They indulge in fatal interactions, and all the reproductive parameters like survival, growth rate, they declines with their increasing density. But there is currently there are no study to show evidence of these, so we are going to look at it with our data. We have information about leopard densities in habitat pockets in Africa, Sri Lanka and Nepal, and through large scale surveys in India. However, information about their demography, we know very little about their demography, and whatever information is available is currently limited to the African and Indo-Chinese subspecies and also constrained by small sample sizes. Indian studies are mostly deals with conflict, space use patterns, dietary spectrum as well as density estimates. Their cryptic nature, long lifespan, larger space use and lack of long term monitoring are the major hurdle for the paucity of this information. To address this limitation, we have assessed demographic dynamics of leopard in gradient of tiger density, prey density and human use. In Kanha Tiger Reserve, Kanha is an important tiger conservation landscape in central India. The reserve is composed of a core area which has two river catchments, Banjar and Halon, and then it has a buffer area and a fen wildlife sanctuary. The Banjar area had high prey and high tiger densities. It is a core area devoid of human activities except tourism. Then we have this Halon catchment, it has low prey and predator densities, and it is also a core area where no human activities are allowed. Then we have this buffer area of multiple use forests where community use are allowed, grazing, anti-FP collection etc., but no hunting and tree felling. Then we have this fen wildlife sanctuary where there is no resident tiger and very scarce prey, and this acts as a corridor between Kanha and adjacent surrounding habitats. Overall, these habitat configuration allows us it provides us an unique opportunity to study the demography of leopards across these habitat gradients. We used intensive camera trapping to monitor the leopard population in Kanha using between 2010 to 2024, and we obtained 481 adult leopards. We used the hotspotter and extra compare to identify unique leopards, spatially explicit SCCR models to estimate densities. Bayesian CGS models to estimate survival. Recruitment was calculated using this formula, and turnover was calculated as a proportion of individuals that are replaced from one year to the next, and we use these tools. Now coming to the results, we observed significant growth in the high prey area, about 5%, because of high abundance of prey and high protection. However, we also have recorded, observed growth in the low prey area, because availability of smaller prey are actually supporting this leopard growth. In the other habitats of multiple use forest and the scarce prey areas, there is no growth, population is stable, probably likely due to the lack of prey and high human use. Interestingly, a study done on the same area by Kumar et al. Found that leopards showed 15% growth rate in the initial 6 years, which has reduced to just 5% in our long term observation, which showed that which suggested that top down pressure of tiger on the leopard densities. Same thing can be observed on a larger scale across, when we look at relationship of leopard density, prey biomass and tiger density across India. For example, you can see here that high leopard densities of around 10 occurs at high tiger density, that is around 10 per 100 square kilometer in habitats like Kanha, Pench. However, extreme densities of leopard can occur only when tiger is below 5 per 100 square kilometer. For example, Kuno, there are no tiger. Then we have Panna, Sariska, Rajaji. These areas have extremely high leopard densities. So this is clear that tiger exert top down control on leopards, and also these high density of leopards occur in the protected areas, not outside habitats, outside protected areas. So it shows that leopards goes through the bottom up limitation. Now this is a very interesting pattern in the sex ratios. When we look at the high prey areas, we can see there is a cyclicity in the sex ratios, where every 3 to 4 years there is an equilibrium reached by the sex ratios, while in the other habitats these patterns were not observed. This is probably due to the different limitations of prey and human use, and we will get to know more about this in the next slides. Now let us look at other more insightful parameters: recruitment, turnover and survival. In the recruitment, females show consistent pattern throughout in all the habitats across in the scarce prey habitat, where negligible female recruitment was observed. But in the multiple use areas and the scarce prey areas, there are some level of recruitment in the males. This is probably due to immigration, not because of local reproduction. Similarly, in turnover also we have seen similar trends. Female turnover rates were consistent across the scarce prey area. However, male turnover increases with declining prey. Now this is not beneficial to the population, because high turnover can be linked to increased infanticide, which can alter the sex ratios, and which we have seen in the previous slide. The sex ratios are highly female biased. In the survival also, male and females have similar survival in the high prey area, and female survival remain consistent throughout in all the habitats, and male survival declines with declining prey. Now this reason for this, that males have larger body size, they have higher energy requirements, they move over a larger area in search for food, and does have higher chances of encountering threats from human, tiger, and as well as their conspecifics. Unlike the males, females can survive on smaller prey species, some studies have shown, and female also have a smaller home ranges. However, it is also clear in our data that although female can survive in low prey area, their recruitments are largely affected in the scarce prey habitats. So they are surviving but not reproducing. We did not find a significant impact of density dependent effects in our study in any of the habitat strata. In the high prey area, there is the impact of tigers are limiting their growth. Then in other habitats, there are impact of low prey as well as human use. So the populations still have potential to grow. To summarize the study, leopards are highly adaptable and often perceived as they will be able to survive in suboptimal habitats. However, our study suggests that leopards require abundant wild prey and effective protection for their survival and population growth. Although they can grow in presence of tigers, high abundance of tiger can limit their population. Connectivity of these suboptimal habitats and with the source population is crucial, and the human activities are the most critical limiting factors in the suboptimal habitats. Long-term monitoring is required to understand large carnivore population dynamics and to inform conservation efforts, and controlling human activities is the critical for ensuring long-term survival of leopards in the suboptimal habitats. I would like to acknowledge these.
Thank you, Jayant. I'll now invite Anubhuti Krishna. Anubhuti has done her Masters in Wildlife Science from Wildlife Institute of India. Currently, she is working in long-term monitoring in the state of Maharashtra. Over to Anubhuti.
Am I audible? Thank you, sir, for the introduction. A very good evening to one and all present here. Today I will be talking about long-term population and demographic trends of tigers and leopards in Vidarbha, Maharashtra. And I am presenting on behalf of the project Long-Term Monitoring of Tigers, Co-Predators and Prey in Tiger Reserves and other tiger-bearing areas of this landscape. Vidarbha. This is the easternmost region of Maharashtra and is constituted by 11 districts. It harbors 5 tiger reserves, Tadoba and Dhari, Melghat, Pench, Bor, and Navegaon Nagzira, several wildlife sanctuaries, including Tipeshwar, Umred, and Penganga, as well as unprotected forests in the form of forest divisions such as Brahmapuri and Pandavkoda. Our first major field estimation is that of the prey population, and this is achieved through line transect-based distance sampling, followed by density estimation using the distance program. Over the years, we have observed that there are several high prey density tiger reserves, such as Tadoba and Pench. There are tiger reserves harboring moderate prey densities, such as Melghat and Bor, and the low prey density tiger reserve of Navegaon Nagzira. We have also estimated prey densities for the wildlife sanctuaries and the forest divisions in this landscape over the years, and we have seen that these areas harbor low to moderate prey densities. However, the prey yield is poor in the sense that there are fewer prey species found here as opposed to the tiger reserves within the landscape. Our second major methodology is to estimate tiger and leopard populations. Field sampling is done through camera trapping in 2 square kilometer grids, followed by segregation of images into species. This is done using AI tools followed by manual processes, and then identification of individuals, followed by analysis and estimation of densities through the spatially explicit capture-recapture framework. We have looked at annual and leopard tiger densities over the years across various sites, and we see that we have certain high tiger and leopard density sites such as Tadoba and Pench. Melghat and Bor harbor moderate densities of predators, whereas Navegaon has low tiger densities and high leopard densities over the years. These estimates are obtained through single session SCCR. When I say single session SCCR, I'm essentially talking about camera trapping conducted over 25 to 30 days, and then we estimate populations and each year estimate is independent of the other year. Now I would like you to keep this in mind because I will be circling back to this after a few slides where we talk about looking at population and demographic trends. So before we move into that, I would like to take you through a small journey of various research outcomes that have been obtained through this project. So we've looked at various aspects of carnivore ecology. We've followed 2 females particularly in Tadobandhari Tiger Reserve over 5 litters each, and we have tried to understand survival of tiger cubs under the influence of high male turnover and density. And we have seen, surprisingly, that only 7 out of 25 cubs have survived for these 2 females, which is only a 28% survival. So is prime habitat for some individuals prime habitat for others? We have also followed tigers in the Pandavkada forest division where we have this one female that has raised 6 cubs to adulthood, and we have utilized this opportunity to analyze kill rates, and we have seen, quite to our surprise, that it is actually easier to raise larger litters where the prey body size is larger, such as availability of cattle in Pandavkada, and this may actually be leading to increased conflict in the future. So this is something we might need to look into. We have also looked at existence of land tenureship systems for leopards through turnover rates and mutual avoidance amongst individuals. Moving on to species interactions, we have looked at sympatric carnivores in this landscape, so tigers, leopards, and dholes, and their segregation in space and time. We have tried to understand the influence of density on these interactions, as well as utilize the time to encounter model to understand spatiotemporal interactions. We have also looked at prey-predator dynamics and used the multivariate Hawkes process to understand these interactions, which takes into account indirect effects of the ecosystem, and compared this to the time to encounter model. We have also worked at developing methodologies, especially for the population estimation of unmarked individuals through camera trap-based distance sampling, the space to event model, and the time to event model. We have looked at dispersal and movement ecology in this landscape where 15 tigers have been collared. And living up to his name, there was a tiger named Walker, living up to his name, he actually moved a displacement of approximately 3,000 kilometers. We have also used accelerometers built into these collars to understand various behavioral states of tigers. And lastly, through our various understandings in this landscape, we have been able to bring conservation management action. And so there was translocation of 3 female tigers to NNTR over the past couple of years. And one of these individuals actually dispersed from NNTR to the forests of Balaghat Forest Division in Madhya Pradesh, elucidating a functional corridor between NNTR and Balaghat. And we also realized that small stepping stone habitats are key to dispersal in the central India metapopulation of tigers. Now, drawing your attention back to single session SECR, so where we left off, single session SECR only provides us a snapshot in time. So like I had mentioned, it only looks at a few days within a sampling year, for example, and it only provides a small snapshot in time. But since we have the advantage of this being a long-term project, we can actually perform multi-session SECR. So multi-session SECR allows utilization of data collected over a series of sessions. It accounts for variability between sampling sessions by using it as a modeling parameter. It provides estimates of session-specific density, detection probability, and sigma, and it allows for estimation of lambda, which is the finite rate of increase. And this allows for understanding population and demographic trends. So lambda is centered around 1, so a value of 1 indicates a stable population. Anything lower than 1 indicates a declining population trend, whereas values higher than 1 indicate an increasing population. So we selected 3 sites, Tadobandhari Tiger Reserve, Melghat Tiger Reserve, and Navegaon Nagzira Tiger Reserve. And we performed this analysis across 3 sessions for 3 years, that is session 1 for 2021, session 2 for 22, and session 3 for 23, with lambda 1 indicating the rate of increase from the first to the second session, and lambda 2 from the second to the third session. And we analyzed various models, we built various models to look at multi-session SECR estimates. So coming to our results, we found that both tigers and leopards in Tadobandhari Tiger Reserve show lambda values close to 1, that is they show stable population trend across our sampling years. When we look at tigers in Melghat, we see an increasing trend for tigers with values greater than 1. And when we look at leopards in Melghat, we see that value for lambda 1 shows an increasing trend. But for the lambda values for the second session to the third session, we actually see a declining trend. Now this is quite interesting because we actually later came to understand this is an artifact of sampling because we only had fewer camera traps in the third session due to logistical constraints. we realized that consistent sampling across sessions becomes very important when we try to understand trends through multi-session SECR. Looking at Navagaon-Agzira Tiger Reserve, we see that the value for lambda 1 shows a strong increasing population trend coupled with a corresponding decreasing trend in the leopard population. When we move from session 2 to session 3, we see that there is a slightly lower increasing trend for tigers, which leads to stabilization of the leopard population trend. Now looking at demographic response of leopards to tigers in these selected areas, when we look at Tadoba, we've made plots where we've plotted leopard density versus tiger growth rate and tiger density. We see that we observe peaks in leopard density either when the tiger density is low or when the tiger growth rate is low. So leopards are only attaining higher densities when either the tiger numbers are low or the tigers are declining in certain areas. We also plotted leopard growth rates, and we observed that there is an increasing trend in the leopard growth rates as we go to areas with lower tiger growth rates. Moving to Melghat, we see that at lower values of tiger density, we see peaks that then gradually increase as this is coupled with the effect of negative growth rates for tigers. So again, leopards obtain higher densities when either tiger densities are low or when the tiger population is declining. We see a similar trend for leopard growth rates where the peaks are gradually increasing from low tiger density areas coupled with the effect of negative tiger growth rates. When we come to Navagaon-Agzira Tiger Reserve, however, we observe a similar trend is also observed for leopard growth rate. Now we hypothesize that this is because the tiger densities in Navagaon-Agzira Tiger Reserve are very low, and this allows for leopards to obtain maximum potential and be able to have higher densities as well as higher growth rates across this space. So to conclude, Tadoba showed stable population of both tiger and leopard across our sampling sessions of 21, 22 and 23. Melghat Tiger Reserve showed a slightly increasing population trend for both these felids. NNTR showed an increasing trend for tigers and a corresponding decreasing trend for leopards. Leopards achieve high densities and growing populations only if either tiger density is low or the tiger population is declining. Multi-session SECR allows us to estimate the values of lambda, which then allows us to look at whether a population is growing or declining, and this helps us to understand source sink and metapopulation dynamics within a system. And since the central India population of tigers survives in the metapopulation framework, this becomes important to understand the long-term survival of these carnivores in this ecosystem. We also understand the importance of annual sampling over long time periods. So for highly case-selected species such as tigers and leopards, if we want to look at population and demographic trends and understand trends in time, we need to do annual sampling over long time periods to be able to attain these estimates. And also we understand why consistent sampling is important to avoid spurious patterns such as the one observed in Melghat. Only if we have consistent sampling over years can we use models such as these to estimate trends appropriately. I would like to acknowledge the following people without whom this would not be possible. Thank you.
Thank you, Anubhuti, for finishing quickly. And I now invite Sankarshan Chowdhury. Sankarshan has completed his Masters in Forestry from FRI, Dehradun. Subsequently, he joined very briefly Bombay Natural History Society, and then he joined Wildlife Institute of India. He has largely worked in Central Indian landscape. Over to Sankarshan.
Thank you for the introduction, sir. So good evening, everyone. So today I will be discussing about if the pattern of mammalian species richness can reflect the biodiversity conservation actions in a human-modified landscape in central India. So this is a part of the larger project named the Development of Landscape Management Plan and Monitoring with special reference to the Ken-Betwa River Link project in Panna Tiger Reserve, Madhya Pradesh. So Panna Tiger Reserve, so we all know the excellent conservation effort for tigers, how the population increased from 0 to now the 70 to 80 individuals. But now in the recent past, the Panna has experienced a lot of dispersal events. At least around 40 individuals have already dispersed from PTR to the landscape, and now it is for us, it is for for us, it is the high time to focus, to shift our attention or to extend our vision from the PTR to the entire landscape in order to increase the conservation value by taking care of the other mammal species. So I would just briefly introduce the present study, the context of the present study. So first, the Asia Pacific, that is one of the most biodiversity threat prone areas, mainly due to the land use change and the direct exploitation of the resources. So actually we need to know, before any kind of policy making, what are the drivers of the biodiversity threats. Now the question is how, how to prioritize the conservation actions. So basically we may have to focus on a single species based approach, or sometimes we can also focus a multi-species based approach. Both are right in their places, but there is a paradigm in between this single species versus multi-species approach for the conservation planning. But largely we believe that it depends on the context, situation and the habitats we want to protect. So now what are the things we need to look at before planning any kind of conservation actions? So the differential pattern of species space use, that is largely governed by the two factors. One is the ecological requirements of the species, and the other is the anthropogenic factors, and altogether these two things are broadly shaping the distribution of the species. In addition to that, the life history traits like food habits and the body mass can also shape the spatial distribution of mammals. It can also provide insights, the trait-specific habitat selection that may extend the vision of a conservation practitioner in order to identify the conservation priority zones. But there are some constraints as well. So species, while working in a human-dominated, vast area of human-dominated landscape, species distribution data are often sparse, and it suffered due to a lot of things like the logistic constraints, the species rarity and their elusiveness, and as well as the imperfect detection. So hopefully there are some models that can handle a few, at least some of the issues I mentioned here, such as the multi-species occupancy model can handle the imperfect detection and also can model the occurrence of rare and elusive species. So to sum up the introduction, so while single species approach, we all know that it may be one of the most widely used or applicable methods, but in a larger landscape where we want to protect multiple species, or we want to have a different varieties of species groups of based on their traits, a multi-species approach can also be adopted. So the objective of this present study is to assess the community and species occurrence in a human-dominated landscape, and we also like to further quantify the responses of different functional groups to the spatial variations of environmental and anthropogenic covariates. So coming to the study area, so the Greater Panna landscape is the study area which has three protected areas, Ranipur Wildlife Sanctuary, Nauradehi Wildlife Sanctuary, as well as Virangana Durgavati Wildlife Sanctuary, and 11 forest divisions. The total forest area is around 12,000 square kilometer, and we are excluding Panna from this current analysis. And these are some of the pristine and serene photographs of habitat shots in the GPL. But let's talk about the human dimensions of the area. So there, the 63% of GPL is human modified landscape, and the people living here are poor. They are very poor, and the Niti Aayog has already labeled this zone as a backward or human development poor zone. So this can certainly indicate the inevitable natural resource dependency on of the people. So coming to the methodology, so we did the camera trapping survey in the project period. We have deployed 1463 camera traps in 4 square kilometer grids, and the data obtained from the camera trap was sorted species wise Then we constructed a detection history, a three-dimensional detection history comprised of the species, site, and the occasion. And finally, we developed the multi-species occupancy model. The good thing of this model is this model first estimates the community effect, then it draws the information from this community effect and determines the species-specific effects, and that's how it can deal with the rare and elusive species occurrence. And at the same time, it can also handle the imperfect detection. So what are the covariates we chose? For the detection process, we used the terrain complexity at 30-meter scale, the effort, number of camera trap days were active, and the protection status of that region, whether it's a protected area or a territorial forest division. And for occupancy covariate, we used forest cover, shrubland, terrain complexity, and water distance as environmental covariates, and human modification index, population density, and road density as the anthropogenic covariates. So coming to the results, so around 43,000 trap nights of effort yielded a total number of 28 species in Greater Panna landscape, with a site level species richness varied in between 2 to 15. The community response, the first thing is the detection. So we found the effort or number of days that for all the species, it significantly and positively influenced the species detection. We also found more number of species inside the PAs and in compared to the non-PAs, non-protected areas. Coming to the community response of the species occurrence, so we found 3 environmental covariates: forest cover, shrubland, and distance to nearest water source, and 2 anthropogenic covariates, human modification and population density. They significantly influence the distribution of these 28 species. And we have also categorized the 28 species based on their diet. So 4 categories: strict carnivore or obligatory carnivore, flexible carnivore or facultative carnivore, omnivore and herbivore. And based on their body mass, we have 3 classes: large, medium, and small. So we found that large and small-bodied mammals with herbivore, strict carnivore, and omnivore diet, they actually preferred forest cover and areas which are close to the water body, but they were also sensitive to the human modification and the population density, as they were negatively correlated. So if we talk about the medium and small-bodied mammals with carnivore, herbivore, and omnivorous diet, we found that there is a variable preference for forest. Not all the species were forest dwellers, and some of them avoided the forest as well. And they showed an equally highly variable response to the human disturbance as well. So coming to the species-specific responses. So forest cover and water availability, they largely govern the overall species distribution, but there are also some species which did not show any kind of affinity towards the water availability. And talking about the anthropogenic factors, human modification and population density, both of them influenced negatively the species distribution, but there are also certain species that did not show any kind of neither avoidance nor preference for this the anthropogenic disturbance. So coming to the conclusion and management implications, so we found the high number of species inside the protected areas. So straight away we can say that the declaring of new PAs or status upliftment of existing PAs can be implemented, or we can enhance the protection, especially for the forests which are outside the protected area. And as usual, the forest cover always governs the species distribution irrespective of their status, but especially if we provide the protection of forest cover in the outside PAs, that will be very much helpful. But one thing which I would like to mention, the maintaining the habitat heterogeneity will be crucial not only for the forest dwellers but for other species as well, especially for the medium and small-bodied mammals like chinkara, wolf, and fox, etc. So habitat heterogeneity irrespective of the protection status should be ensured. we showed a substantial variation in response to the human disturbance. So some large-bodied mammals with carnivore and herbivore diet, they showed a kind of preference, and some of them also showed, like tiger here, avoidance from the human disturbance. But there are some small-bodied mammals also there. They showed an attraction towards the human settlements. So this kind of variation indicates there is a possible human-wildlife interface is at play in GPL, which can lead to the conflict with people in terms of the livestock depredation, as well as the crop raiding, and also it evokes the need for monitoring the health of wildlife and human health. And also the linear infrastructure, the last thing I would like to mention, because for some species we found they are very sensitive to the road densities. So such as for tiger, because we found the tiger occupied areas are actually traversed by many roads. So for that we also think about some kind of suggestions or recommendations. So I would like to conclude my work by mentioning the development of our project. Like, we have already prepared the integrated landscape management plan, and that has been submitted to the Government of India. And here, and also which is in line with our present study findings, the 2 tiger reserves, the newly declared tiger reserves, this is the status upliftment of 2 wildlife sanctuaries. One is Ranipur Wildlife Sanctuary and another one is Nauradehi Wildlife Sanctuary. So both of them have become the tiger reserve, and there are another 2 or 3 community reserves and conservation reserves are yet to be declared. So along with some of the findings with this present study and along with other recommendations mentioned in this plan, so we hopefully we can say that in the next 10 years, all these recommendations will be implemented in the Greater Panna landscape for the overall conservation value. With this, I would like to acknowledge the following people for their institutional support and the field support, and thank you very much.
Thank you, Shankarshan, for finishing quickly. Now I invite Priyanka Justa. Priyanka is MSc student of Wildlife Institute of India, and she is currently pursuing her PhD focusing on high altitude carnivore species in Spiti. Over to Priyanka.
Thank you for the introduction, sir. What comes to mind when we picture Trans-Himalayas? The rugged terrains with snow-capped mountains, and elusive species like snow leopards and Himalayan wolf. Yet, amidst this breathtaking beauty lies a complex narrative of survival and competition amid encroaching human presence. What happens when these native predators are forced to share their habitat with rapidly increasing human activity and their domesticated counterparts? Good evening, everyone. Today, I'll be talking about how human-altered landscapes are reshaping carnivore niche spaces in the cold desert of Spiti Valley. This research is a part of a 3-year project titled Niche Selection and Mesopredator Release in High-Altitude Ecosystems, supervised by Dr. Salvador. So last year in ARS, I mainly focused on interaction pattern among predator guilds, so basically focusing on entire predator guild in this region. And we found moderate to high diet overlaps, and we saw that spatial interaction among these predators is governed by various covariates. We found that spatio-temporal niche partitioning plays a key role in their coexistence. And notably, we identified free-ranging dogs as a crucial part of predator guild. So this year, I'm shifting my focus, specifically focusing on dog interactions with native carnivores. We'll also examine the changes in dog populations over the past decade. And this analysis has become more robust by adding new data sets. And further, we have statistically analyzed niche overlaps, so moving beyond just the overlap coefficients that I presented last year. So to set the stage for my presentation, let's revisit how predators interact. So carnivores, they compete for resources aggressively, forming trophic hierarchies that shape structure of an ecosystem. So to reduce competitive interactions, predators they try to avoid competition by partitioning their niches across diet, space, and time. Now, here's come the twist. Usually, these competitive interactions are governed by body size, where the larger carnivore dominate the smaller carnivore. But these interactions can be modified by humans as they have varying impacts on different carnivores. For instance, there are some opportunistic species that thrive very well in human-modified landscapes by taking shelter and depending on anthropogenic subsidies. So in this way, humans indirectly, they can help increase the population of some carnivores, and indirectly they can it can lead to intense competition for the other carnivores, which avoid humans. This human shield effect is particularly relevant in Trans-Himalayas of northern India, where the region has experienced a tourism boom in last 2 decades with expanding human settlements. So while tourism is good for locals, but it also brings the problem of unmanaged waste, which attracts opportunist carnivores like red foxes. But this unmanaged waste and subsidies, they can support another carnivore, which is a introduced carnivore, the dogs. So they can thrive even better than red foxes. And the growing dog populations, they can often outnumber native carnivores, intensifying competition with native predators and putting pressure on an already sensitive ecosystem. So these free-ranging dogs, they can outcompete the native mesopredator, which is red fox in this region, and even they have the potential to outcompete the top predators in the region, that are snow leopards and Himalayan wolves. So why study interactions in the region? It's because previous studies have focused individually on these species, both the top predators and the mesopredators. And while in the broader Trans-Himalayas, the studies have been done on interactions, but they have either focused on one aspect, or they have not taken into account the free-ranging dogs, which have become a major part of this predator guild. So there are very few research that have looked in interaction patterns in the region. Identifying these research gaps, our research had 2 main objectives. First is to estimate dog populations in some villages in the Spiti Valley and assess the decadal changes in their population. Second is to examine intra-guild interactions between dogs and native carnivores across spatial, temporal, and dietary niche axes. We hypothesized an increase in dog populations over the past decade. Then we also hypothesized there will be a high dietary overlap between dogs and native predators, intensifying competition for limited resources. Further, we expect that these native predators will be coexisting with dogs by separating their niches across space or time. Our study area is known for its unique biodiversity and extreme temperatures. The vegetation is alpine scrub. Community is majorly agro-pastoral, but tourism is increasingly becoming an important revenue source. Sheep, goat, donkey, zoo, which is cow-yak hybrid, these are major domestic animals. And so within the entire landscape, we selected few intensive study sites based on a disturbance gradient, considering tourism impact, number of villages, livestock, and dog numbers. These sites arranged from the least disturbed and two most disturbed are Gaur, Chandratal, Pin, Mane, and Kibber. So we conducted our study in 3 sessions. In the first session, we deployed camera traps in a grid of 1 by 1 kilometer square, intensively covering all these study areas, followed by a third session where we covered entire valley in a 5 by 5 kilometer square grid, covering the extensive area. And dog population estimation within the area was carried in Mane, Pin, and Kibber study sites. For dog population estimation, we overlaid 1 by 1 kilometer square polygon, depicting an average home range size for free-ranging dogs. We conducted foot surveys and took dog photographs and locations. And we used SECR to estimate their density and population, where lambda was the detection probability, represented detection probability, and sigma the movement scale of animals. For villages with fewer dogs and fewer households, we deployed block count method. For diet analysis, we collected scat samples over all the sessions. So in addition to these samples, we also included some previous samples from already published literatures, so that we ensured that we had minimum of 250 samples per species. So first we scraped outer layer for DNA analysis to ensure that the species we have identified is correct. This analysis is still ongoing. Then followed by it, we separated undigested items and identified them. And prey species were identified using medullary patterns. We computed relative frequency of occurrence for each food item, and then analyzed dietary niche overlap using Pianka's index, with further testing whether the observed overlap deviated from null hypothesis, which assumed independent food item consumption. For spatiotemporal interactions, we used the camera trap data. For spatial interactions, we combined session 1 and session 2 data and used multi-species occupancy in R using covariates like elevation, terrain ruggedness, etc. Then we analyzed temporal interaction patterns using calculating temporal overlap index and statistically comparing it with the overlap over detections distribution across dial cycles using Mardia-Watson-Wheeler test. Coming to results. First is dog population estimation. So here I'll only share the results of Kibber study site, because comparative data set is only available for this study site. We see minimum of 11 dogs in Langza to 60 dogs in Kibber. And further, we see that in all these villages, a notable dog population rise has happened from a percent increase of 87% to an astounding 562% in Chincham. This increase is probably because it is governed by the village size and livestock number, as has been seen in the previous study. Coming to the results of density, we see dog densities ranging from 17 dogs per kilometer square to 59 dogs per kilometer square, which is a very high density, considering especially that the native carnivores, for example, snow leopards, have the densities of, in this region specifically, 2 snow leopards per 100 kilometers square. So seeing this disparity, it's obvious that the dogs are increasingly becoming a major part of this predator guild, shaping the ecology in this region. One important thing to note is that we are still doing the analysis for Kaza, which is the main tourist hub. And here, the initial analysis show that we have a head count of 312 dogs, 312 unique captures, which is up from 224. So again, a very high increase in the population. So now that we have established increase in dog numbers, we see dietary habits of carnivores. We see that for dogs, cattle forms a major part of their diet, and human-derived items also form the second most important part. So, but overall, livestock forms a major part. When compared to foxes, foxes have small mammals which contribute majorly to their diet, like rodents and logomorphs, but cattle also forms a major diet, which is likely attributed to this scavenging behavior, which is a successful mesopredator strategy. But we see that human-derived items are lesser compared to dogs, which has been seen in previous studies that where dogs are present, the share of human-derived items decline in red foxes' diet. Further, snow leopards had a mix of wild prey and domestic animals in their diet, and we see highest proportion of domestic prey in the diet of wolf, which might lead to human-wildlife conflict in the region. So this represents the comparative diet of, this is the observed utilization of diet, and we computed dietary overlap using this and compared it with expected utilization to see whether the diet overlap is significant or not. So we found that the diet overlap is significant for all, like for dogs with all these predators, mostly because all of them had a fair share of large and small bodied domestic animals in their diet. So seeing that there is a high diet overlap, we moved to study their spatial co-occurrence patterns. Broadly, we see that these log odds of co-occurrence increase with increase in elevation, implying that dogs have even ventured to highest of elevations, which are commonly associated with high altitude carnivores. We see the co-occurrence decline with terrain ruggedness given habitat constraints. We see that with given prey covariate, dogs co-occurrence increase with red foxes and decline with snow leopards. We further see the co-occurrence increase in area near roads and decline near human settlements. So basically, log odds of dogs co-occurring with these predators increase as we move away from human settlements. Then overall, the positive co-occurrence is harmful, like it raises concerns because they have ventured into highest elevations and they are co-occurring in space. So maybe potentially they are creating opportunities for disease transmission risk, which needs further investigation and competition and hybridization in case of wolves. Finally, the temporal overlap. While these predators overlap high in diet and space, we see they separate in space, like temporal niches. So dogs, again, they may be restricting the resource use of red foxes, leading them to avoid dogs. And we also see that dogs and the top predators also separate themselves in times. So basically, dogs were found to be diurnal and native carnivores, nocturnal or crepuscular. So human activity and provisioning, it's known to influence dog behavior, but they together may be constructing the temporal niche of native carnivores. So to summarize, we have seen a notable increase in dog populations. We see high dietary overlaps, implying competition for limited resources. And so these predators, they are able to coexist and by spatial or temporal niche segregation. We recommend long-term monitoring of both predators, like native predators and dogs in the region. It would be interesting if few individuals of all these predators, if we will be able to collar them to study the fine-scale space use of these animals and how far these dogs are moving. And lastly, the dog population needs to be controlled by multi-pronged methods using waste and carcass management. So that's the take-home away message, that dog populations, it's high time that these populations be managed in the landscape. So with this, I would like to acknowledge these people. Thank you, Julie.
Thank you, Priyanka, and I'll hand over mic to Chair.
So, thanks a lot. Perfect timings. I'll speak later on. We'll first take questions. I'll ask Vaibhav to take them.
All the speakers, please come up.
Okay, should I read it out? So, thank you, sir, and good evening, everyone. Quite a few questions, obviously an interesting session. So the first few questions for the first presenter, Dr. Bilal. This is from Khushi. Can spatial capture-recapture models for group living species like elephants be reliable to consider both groups and individuals, solitary males in brackets, for population estimation?
Yes, that's a very, very interesting question. So what we did, we actually, when we identified that there are some solitary males, we actually removed the solitary males. We considered only the males which were part of the group and then tried to estimate the population. Because solitary males in any given area, in any given population in elephant reserves, there are very limited solitary males, like you can have 5, 10, and most of the people identify the solitary males, so you don't need to do anything for them.
So the second question for you is from Dr. Ramesh. How was the design determined for each method, and did you compute effective sample size required for each method? Even with a low CV of 24%, why is confidence interval very high for some methods?
Sample design in the sense like we try to have a full area coverage so that there is no misses. Now what we are trying, we are actually working on 3 different aspects. What is the precision versus bias? You may have a very good CV, but you may be totally biased. You may be precise, but you will be totally biased. Your estimates will not be good. But you may have a very high CV, but you will not be the precise. So that's what we are trying to evaluate and trying to see which method actually works good when we are talking for the estimates, when we are talking for calculating, using the method for the population estimation.
Thank you, Dr. Bilal.
Bilal sir, I have one query because nobody's asked that. Are these sampling done at the same time together, or they were spread at different times? Because the elephants move a lot.
Sir, all together except dung decay, because dung decay go for a very longer time.
Okay.
But rest all the sampling done at the 45 days at one time.
Thanks.
Thank you, Dr. Bilal.
Thank you.
Question for Jayanta now. This is from Prashant Mahajan. Nice presentation. What ecological role leopards may serve in high tiger density areas, even if leopard densities are high/intermediate?
Thank you for the question. Definitely leopard can, in presence of an apex predator like tiger, they can play a role of major predators. They can prey on langur, and all the food habit studies done in India, they suggest langur, hare, all these small carnivores, they contribute a significant proportion of their diet. Thanks.
So, thank you. That was just one question for you. Question for Anubhuti now. This is from Omkar. What is scale of high and low prey density, or what are the densities if the prey base in Tadoba, Melghat and NNTR? Is it considering only wild prey or livestock as well? As per TATR 2019 reports, the prey density, specifically Sambar, Cheetal and Gaur together is around 15 per square kilometer.
So when I talk about high and low prey density, I am only drawing comparison within the various sites within Vidarbha, the sites that we have looked at. And the values I had presented were only for the wild prey. We have not looked at values for cattle as of now, and I have not presented those values. And so the values that I had presented were for individual prey, but then taken together, like you're mentioning, taken together, we can also look at overall availability of prey. So that is the basis which was used to compare them with each other in terms of high, moderate and low prey densities.
So the second question for you, rather fundamental, by Dr. Johnson, whether tiger and leopard are K-selected species or R-selected species. If it's K-selected, please explain the qualifying characteristics.
So sir, they are K-selected species since they have life history traits which evolve over long periods of time. So lot of time between births, lot of time for age to first mating, age to first reproduction. So,
sir, can I just clarify, sir? For example, leopard, it is a versatile species. It's not a habitat dependence one. Okay, every alternate years, it gives litters, and they have very good parental care. Okay, and every alternate year, even the cubs can ready to go for reproduction. So that's what I have this question, whether it qualifies as a K-selected or R-selected species. If it is K-selected means, then what about rest of the species which have highly specialized in their habitat. Okay, thank you anyway.
Okay.
Yeah, thank you. Question for Sankarshan now. This is from Dr. Bilal. What happens to occupancy assumption in multi-space modeling considering variation in body size? And the second part is, was 4 square kilometers grid size for all sizes?
So, based on the body size, like the occupancy assumption, I mean, in our study, we have actually relaxed the occupancy assumption because there are 28 species involved. So, they have different body sizes and they have different area requirements. So, we have already relaxed that occupancy assumption from occupancy to the probability of site use. But this multi-species occupancy modeling, this actually calculates the species richness. I mean, the assumptions would be broadly the same, but the calculation or their final output will differ. So the species richness, as well as they also produce the occupancy as well. So the thing is, we have already relaxed that assumption for multi-species occupancy model. And the second question.
Was the 4 square kilometer grid size same for all sizes?
Yes, and I mean that actually also comes from my the previous answer. Yes, the 4 square kilometer grid, and I would say the inter-trap camera trap distance was around 2 kilometers on an average in the entire landscape. Yes, that is also for all the species, and there are multiple evidences that we can use, or we can use the smaller areas or smaller grids or points in compared to their home ranges to estimate the species richness. Yes.
Right. Second question for you. This is by Akanksha Saxena. Great work. What is the underlying hypothesis behind considering that animal dietary habits would be reflected in or related to habitat preferences / suitability? And the second part of the question, which is as on date not very politically correct, what are the possible implications of your study for the Ken-Betwa project?
Okay, first the underlying hypothesis of the body size or the food habits, the life history traits. So the body size and the food habits, like they actually govern the space use patterns of the mammals, because here we have seen the larger body size mammals with carnivore or strict carnivore habit, they will prefer contiguous forest patches. They will require more amount of water. They will also be very much sensible to the human disturbance. So and they also, I mean, the large body sized mammals, they also have a longer generation length. So these traits actually, and these are actually governing their space use. So when we are incorporating this information in our modeling, so that will help us to identify for, the resource selection patterns in a better way in compared to the other methods. Other methods means if we do a species specific response, then also we find some amount of evidence in line up with this. But the group specific or life history trait specific response will give you a more accurate estimate or more accurate interpretation. I mean, what area to conserve, what area not to conserve, something like that. And the second question is like whether it is related, how it is related to the Ken-Betwa. Yeah, I have mentioned in my last slide that we have already submitted the integrated landscape management plan. This is the part of that integrated landscape management plan document where the since you already asked this question, the harmful effects of the Ken-Betwa river link project, to mitigate those harmful effects, there are a lot of species specific and theme specific recommendations have been given. So my study here lines up with the spatial conservation prioritization of the GPL landscape. So that is how it is linked with the my study with the Ken-Betwa link project in Panna Tiger Reserve.
Right, thank you. Questions for Priyanka now. What are local people's, this is from Yash, what are local people's perceptions with increase in dog population? And second is with dogs hunting, consuming livestock.
So local people in some areas, they have, even like in Pin Valley, they have just, they told me that they have left rearing these domestic animals because of dogs, because they kill livestock in such huge numbers. But given their religious beliefs, they won't allow maybe culling, or they won't favor that.
Right, the second question is from Dr. Bilal and Varun Kher. Comparison of dog density from smaller areas with snow leopard density at larger scales may not be appropriate. So what are your thoughts on this?
I don't think why it's not appropriate. I mean, I cannot agree with you, because even I don't know. I mean, do you think that it's not, you will be able to say that dogs are not a concern given like such high densities? Even if one snow leopard gets like hurt, I mean.
Question is like the dogs that you have shown is a very small area, and the snow leopard habitat is a very large. So the impact of these dogs, the smaller village, will be up to some distance. So they may be affecting 2 snow leopards, 3 snow leopards.
Yeah, but
they will not be affecting the entire population, because so you were saying the 54 dogs per square kilometer, and then you compared it to 0.2 snow leopards per square kilometer. So that comparison is not logical because it's a huge landscape versus a very smaller area, because dogs are fed by the people, dogs are around our villages, they have an impact on the wildlife, but that is limited to some distance. So that's what we were trying to ask. So upscaling it to the entire, we will say that all.
No, I just compared the Kibber landscape and Kibber snow leopard density with Kibber, and this is not just one village. Like these,
exactly.
I just compared village wise,
That's
but
what we are
if
saying.
we combine these villages, then the impact will be even greater.
But landscapes are much larger, so its snow leopard density will occur only at that density.
Bilal,
Dogs-
sir, can we discuss this separately?
sure.
Thank you.
That is all. Thank you very much, sir. Over to the right. So hearty congratulations to all these speakers. Wonderful presentations. The theme of coexistence in this session was rather between tigers and leopards rather than humans and wildlife. So that was a kind of a digression from the theme of the ARS. But anyway, very well done. I was but hoping for a finality to the method which needs to be adopted for elephant estimation in the country, because I was myself at the receiving end when I was in the field. I had 3 numbers from 3 different methods. One was done by Vishnupriya, one was done by Ujjwal from Tigersal, and one which we did ourselves in the department. So I didn't know which one to project. The best figure was Ujjwal's from Line Transex, but it was a remarkably high figure which I could not trust. So I'm hoping Dr. Bilal and his team would arrive at some kind of finality which we can adopt at the ministry in due course of time. Because with such variations in the process itself, it's very hard to comment whether our populations are stable or increasing or decreasing. So I'm hoping that that would happen soon. Similarly, the same approach needs to be adopted for rhinos also in the country. Since I'm from a rhino-bearing cadre, we have faced this problem in context of rhinos also. We have tested multiple methods in the field where they gave similar results. The differences were not as high as in the case of elephants. So I would request Director, Wildlife Institute of India, Tiwari sir, to look at the possibility of evolving a countrywide standard methodology for rhinos also. So very well done, all speakers. Thank you very much. Over to the chair, sir.
Thank you, Weber. I think, as usual, a perfect session. I would congratulate each and every presenter. Excellent studies, very extensive areas covered, and very long-term data collection. And I think I was bowled over by the analysis because half of it went over my head. So My request is please, in the end, if you can give very simplified analytical results for the managers. You know, we managers in the field, it will be good for your impact assessment journal or publication, but for somewhere you should also give a very clear-cut understanding, so that, because you know, you are looking at the landscape, and that's where we are also trying to work out, and these studies are going to complement our management practices. So I always keep that in mind. For paper writing, very fine, AR is very fine, but somewhere you should also give these research findings in a very simple articles which can be shared across with everyone. So even a common man can understand those things. So that's my one advice. And perfect answers, you know, you are very confident. That's the best part, because this is part of your training. You are doing research and you are the next gen. So you will take over from most of us over a period of time. And I think this is perfect. I really like the way you answered, the way you reacted. Perfect. Thank you so much. For this part, reaction part. Coming to Bilal Saab's this thing, it was a case like a room in an elephant, which is Mr. Bilal, Dr. Bilal himself, to the missing elephants in the room or in the PAs. So, as Vaibhav mentioned, yeah, these are serious things. The research is good, findings are good or bad, but how research, this critical part of analysis has to be, you know, these CVs and all are given, but then other studies and other comparisons should be very, very clear. And I think Vaibhav has already raised that. We need to look into this missing elephant, not only for the elephant, because the landscape is complicated. As a DFO, my first posting in Andamans, we had feral elephants in Interview Island, and we called best experts of that time from Karnataka to capture those elephants. After few days inside the forest, they said, "Sir, it's not possible. We can't see them. Where are they?" But they were there, all over there. It's a very dense forest. So it depends on the landscape, it depends on habitat and all these things. The elephants, Africa is fine, but then we have different landscapes. Some of it is very dense, or terrain is also quite complicated. I have worked in Meghalaya Gaur Hills. The landscape, amazing landscape. We've got Grand Canyon, the small Grand Canyon over there, and the elephants' movement is amazing. So it's not very easy to work and study in these landscapes. Even for a big animal like elephant, then when you look for the smaller ones, things become more complicated. So every researcher should keep that in mind and keep on testing your things in the field. And if you have got any doubt, please mention it critically, so that it helps the managers, it helps the other people who are going to use your results. Thank you so much. One thing I got in the Central Indian landscape studies, the results for 3 studies are pointing towards the same sort of final analytical aspects of these carnivores and how these big carnivores are behaving or are supposed to behave. The approaches were different, the techniques were different, but then the results are actually quite similar, more in some aspects. So I would request if you can generate a combined picture using these long-term studies. I think that will be one of the best thing which will happen on such a large landscape. It's actually covering the entire central India, if you include Kahana, Panna, Vidarbha and all those things. So it's beautiful. So please look into that. Please come together, all you 3 group of researchers, and then try to put it in a much bigger picture in a more analytical way. And that will be quite interesting. The last presentation was perfect. You know, I said we had charismatic animals, but if you are looking at the dogs, yeah, they are creating impact on various landscapes, not only in Ladakh, but we have come across in our beaches where turtles are nesting and things like that. So yeah, these are real critical problems, man-animal conflict, our impact, human impact on the landscape and how we can mitigate them. And these are very, say somewhere it's emotional issues, some are cultural issues, somewhere they're political issues. So we have to be careful also, but then such studies are required. Big animals, well-known animals, very fine, but these are very, very important part of the missing links of our entire picture, and I'm very happy with that. Thank you for that, and please keep it up. All the best, and thanks, Kamar, for conducting the whole thing. Thank you.
Sir, just stay back for a moment. Thank you so much, sir. May I request Dr. Satyakumar to come and give the gifts to the chair and the co-chairs. Sir. Thank you, sir. With this, we come to the conclusion of technical sessions for the day, and how much late we are, doesn't matter. So we have the most interesting thing coming up for the evening. We have a film to be screened, and the film, I won't say anything about the film. So I will invite the star of the film, no other than Dr. Parag Nigam, to come and brief you about this film. This will be followed by, I believe the filmmakers are here. They will also talk to us, tell us briefly about the film, and then we will have the screening of the film. After we finish screening the film, we will go for dinner.
So very good evening, everyone. It's been wonderful to see the whole day being really jam-packed and to still see all the audience in. And this is something very special to us. And really thank everyone to be there for the evening. It's been a long day, but it's going to revive you back. We've had a good amount of theories, good amount of science, and now let's go back to the field. And in this regard, we have the Madhya Pradesh Forest Department had, this got this film made, and WI was a technical partner into this whole setup. We were the scientific advisors to the department, and this is one of the success story which we feel now. We're very confident things have been really going on well. We started our journey way back in 2010 and 11 when the initial Gaur captures were done, and that was basically from Kana to Bandhavgarh. The documentation, we moved 50 animals that time also. Documentation was less, but then that was the first time, a lot of anxiety. And on a local level, on a very simple level, we made a small clips. And then things started on. We studied those Gaur for the next 10 years. And suddenly we had a great leader, Mr. J.S. Chauhan sir. And he said, let's move and revive this whole thing again. And we understood that we had the strength, but then we should not be losing that strength. And then we got the team together. And this time we had really beautiful stuff because it was all Indians doing that. Initially we had South Africans really helping us. In this regard, we had very famous conservation manager, Mr. Les Carlisle, the Sand Park and Umfulozi doctor, Dr. Dave Cooper, Jeff Coke. These are people who really contributed and gave us the strength to really, do things on our own. And we really proved that. Lot of confidence with WII. Really thank the Madhya Pradesh Forest Department for trusting us. And we have a small team. Dr. Bilal is there, Ritesh Vishkarma, Bhaskar Bandari. And now we have another, now Gaur is becoming really big. So we have another calf also joining our team. We have Arun Gorati also is joining the team of Gaurs. So this is going to be a story of what went in, how things were happened up in the field. And we really feel proud that yes, we could do something for conservation, reviving an area, bringing animals there. And now we've gotten a lot of this Gaur translocation has become a big thing. many other states have been asking about, moving animals and say, let's plan mega herbivore translocations, and the strength is there with WII and the local institutions also. So it's not just this film, but it has really brought in a change at the country level, with the Andhra Pradesh Forest Department taking now a lead with the beautiful work on Nagarjun Sri Salem. They want to bring in Gaur back. Dr. John Singh, he wrote about bringing those animals back. Karnataka has been taking big lead, and now we have Chhattisgarh also taking a lead onto that. And to an extent, even the government of India plans to carry these animals to Sri Lanka and further. So it's really brought the Indian conservation picture at a bigger level. So I have a very special two people who have really joined us, especially for this, is Mr. Nishant Kapoor, who is the filmmaker, and we have also Ms. Himani Singh Chauhan. She's a little shy, but then I would request, it will be nice after this, Bivash, now or later? So okay. So I request both Nishant and Himani to join us on the stage, and would request our director to kindly felicitate them, and then we start with the film, and that'll be wonderful. Acha, come. Okay, yeah, sorry. So let's hear also from the filmmaker. Few lines. He'll also talk about his experience, please.
I've forgotten all my lines. I'm a bit nervous right now, but just very, very fortunate to be here. And we work with a small team. We try to bring conservation stories to the masses, which I believe is very important in today's day and age. So yeah, thank you WII, and thank you NTCA and the Forest Department of Madhya Pradesh for this opportunity.
Special moment to turn gift for both of them. Who's helping me out? Start whenever. Okay, lights को थोड़ा off कर देंगे. And this is all make in India thing. So we've got all. हाँ, कुछ नहीं करना है.
12 rivers. Over 30 tigers. More than 4 lakh acres of forest, grassland and perennial streams. Sanjay Tiger Reserve has it all. Except something that was found here earlier has been lost. Gaur.
I have been working here with Sanjay for quite long, and we were always thought the diversity has to be there. What is missing? So yes, Gaur were definitely missing.
With a fully grown bull weighing over a ton, the Gaur is the largest species of wild cattle in the world. Once abundant here until the mid-90s, in just a few years, the last Gaur had disappeared.
From entire landscape from 95 to 2000, all the Gaur gone wiped out from here.
But the Gaur is the custodian of the grasslands and an important link in the ecological chain.
And Gaur is a mega herbivore, and you need a force feeder. Basically, that's how the whole natural cycle operates.
Now, over 20 years since its disappearance, a team of passionate officers has set out to restore Gaur to Sanjay Tiger Reserve. But it won't be easy. The team must pick and move select individuals, which means they cannot capture them by herding an entire group into a corner. Instead, each Gaur must be chemically sedated and manually lifted into a truck.
Capturing tigers is a very different technique. It's a simple stomach, it's not like mega herbivores, they have 4 chambers. You have to take care of them.
Some of the animals are extremely large in size, weighing all the way up to 1,000 kgs.
In this quest, the team will need to employ skill, strength, alacrity. They'll need to brave the weather, survive injuries, and suffer heartbreaks. All to repatriate the top bovid to the home it once abandoned. Success is not a given. Risks loom large. But the hands of these heroes won't rest until Sanjay welcomes the return of the giants. Relocating Gaur is like relocating trees. A task that demands as much delicacy as brute strength. So the team must be sure why it needs to be done.
Main objective of moving Gaur to Sanjay was to ensure that you have a rich biodiversity restored in the park. They shape forests, they shape grasslands, and that's how the whole system works. They're important prey species for tigers, and Sanjay is the one which is having a recovering tiger population. And it's very, very important for us to have them back into system.
Gaur has its role because Gaur used to be there earlier. And so we are just putting them back in their original ecological role.
Fortunately, it has been done before. So while challenging, the project isn't unprecedented.
The Gaur translocation we had actually undertaken way back, maybe 11 years back, from Kana to Bandhavgarh. Basically wanted for a long, long time, and it's happening now.
We have had one of the warmest winters. We have had one of the coldest summers, and the April, May were one of the wettest April, Mays that we have ever seen. In this context, we need to understand that this is really important to do.
The people who had done it the first time, the staff that we had, so we were obviously losing them because they were retiring, 10 years have passed. So we wanted to, this was something that we wanted to continue.
This time we are doing this without help of international veterinarians and capture managers. So, there's a lot of sense of pride in that we are able to do it on our own, though we've got help from international experts in the past. But I think the technology transfer has been very nice, so I think we can do it now on our own.
With a team determined to execute the project in-house, leaders of Kanha, Sanjay and Satpura Tiger Reserves got together to form an elite team. The Forest Department of Madhya Pradesh commissioned the Wildlife Institute of India to provide technical help in the translocation and undertake necessary studies.
So we need to understand that whether if you move animals, will they survive for the next 100 years? Whether habitat is suitable enough. What is the vegetation? What are different structures of the forest which are available? And based on that, this whole map is done. We would also ensure we would have something called as a population viability analysis. That means what number of animals have to get into a new park.
With the suggestion of Madhya Pradesh Forest Department, I did a complete suitability analysis of Gaur in entire Madhya Pradesh, and this is how this project started.
Here when we did the population viability analysis, we said, if you bring in 50 Gaur, and then there would be some supplementations in future, and if you are bringing it, the animals would survive for the next 100 years. So just putting a few individuals will not make any sense. You need to ensure the herd integrity, how males and females interact, what is the birthing interval, what's the gestation time, what's the generation interval, all these factors have been taken in.
With its thriving population and genetically strong stock of Gaur, the natural first source was Kanha. A child of the Satpuras, Kanha is an iconic sal and mixed forest with mist-soaked grasslands. Famously home to the rare Barasingha and a thriving population of Gaur. The task of picking the individual animals for capture went to S.K. Singh, the park's field director.
What will be the quality of herds, how it will be chosen. That was assigned to me and my deputy director. With the help of Wildlife Forensic and Health Institute, Jabalpur, they conducted different tests with animal dung so that it could be finalized that no any disease is available with the herds.
Meanwhile, the team had to take steps in Sanjay to make it a safe haven for its new tenant.
Disease surveillance work was done in Sanjay, and that we got the surveillance of foot and mouth disease, and we also did the sero surveillance of tuberculosis in and around Sanjay Tiger Reserve.
And we are taking seriously the interaction of the cattle with the wild animals, and the vaccinating and making a immune ring around the national park is the way we are securing them.
We had 10-11 elephants here in wild herd, and a few days before they came again from the Chhattisgarh. So that posed us a great challenge to prepare the enclosures. But our commitments and our hard work from our entire staff ultimately gave the results, and we had enclosures for that purpose.
With the team in place, preparation begins in earnest.
These operations are never an individual base, it's always a team effort. I'm really fortunate to be working with a wonderful team. Many of them we have worked together before, but this time bringing everybody together, really great.
We identified the herds, we made teams to track those herds, to keep track on them, so that broadly we know where they are going. We have to have our elephants move into the herd so that the herds are habituated with the elephants, so they don't balk at when we do the operation. Also, we have to scope out the areas where we can do it, where we cannot do it, based on the logistics.
We use narcotics in all our captures, and that's a very tricky thing. It's just a drop is sufficient for animals. So those drug calculations, ensuring thing, you would have sleepless night, a previous night before what you're going to do, how much. So there's a lot of mental stress.
Much will depend on the skill of the vets darting and sedating the target animals.
Basically, the principle is that single dart, single capture. If your dart is perfect, shot is perfect, then your induction time is perfect, then we can safely secure the animal.
So we are putting a VHF and a GPS collar, and we are also preparing the individual dossiers to identify the herds.
The stretcher teams needs to be trained. The equipment has to be streamlined. It has to be built according to the needs of this capture. And you have to be sure that the team you are working with is strong enough to lift animals weighing up to 1,000 kgs.
But the team doesn't believe in plans on paper. Experience breeds confidence, so everything is being rehearsed.
So we have a team of about 25 people. Some of them have been a part of earlier Gaur translocation programs, so they knew exactly what needs to be done, and the others were trained by us.
We did our mock drills because it's a very heavy animal, and there's a specific way in which you have to put it on the stretcher and then lift it and take it to the vehicle. Gradually increase the weight from let's say 600 kgs right up to 1050 kgs. So we were fully prepared for this.
800 kilo बढ़ गए, ठीक है.
All this is grueling work, but the team knows that they must put in the hard miles now if the captures are to run smoothly. The transport of the captured Gaur to Sanjay must be carefully planned too. The vehicles are custom built, with a wide enough sliding door for ingress and egress, ventilators for observation, and fans and mist generators to keep the biochamber cool. All crucial for the well-being of the hooved giants undertaking the most important journey of their lives. The first capture is always the most tense. This is what it all comes down to. All the discussions, preparations, mock drills. The moment of truth is when plans come up against the imponderable. It's time to jump into the deep end of the pool. It's time to beld above it.
So, dart secure. Huh?
चलिए कुछ ज़्यादा बिल्कुल इधर नहीं चाहिए बस ये वाला कितना है ये मुंह ऊपर क्यों उठा रहे हो इतना मुंह उठाना ऊपर उठाना नीचे छोड़ छोड़ SpO2 टेंपरेचर और लास्ट कर दे दूसरा दो
रेस्पिरेशन रेट 10
रेस्पिरेशन वाज 10 तो आराम से निधि कब स्पिरिट वगैरह मत लगाना नो स्पिरिट
निधि थोड़ा ऑक्सीजन लगा निधि ट्यूबर प्लेन का है आई थिंक सर वी ऐसे नहीं डालो नीचे अरे वाले डालो और उसे डालो स्ट्रेचर को आ गया अरे धीमे मेरे पीछे आओ छोटे छोटे कदम आ जाओ सर हेड नीचे डॉक्टर गुरु पीछे ध्यान रखना सबको नहीं जाने की जरूरत है फैलते जाओ निकाल लो पहला निकाल लो उधर जाओ ऑक्सीजन और धक्का दो आप लोग वो निकालो
Remarkably, the first capture is clinical.
अभी नहीं रुको ना जो पहले ये वाला खोलो इसको बंद कर लॉक करो उसको लॉक करो
The team goes right back in for a second capture. And aces it.
जबड़ा नीचे पकड़ो नहीं करना टेढ़ा नहीं करना पाँव खींचो
Buoyed by back-to-back successes, the team pushes for a third.
Capturing herbivores like mega herbivores like Gaur, you have to take care of them. And capturing tigers is a very different technique. Here with the tigers, when you dart the tiger, you need not to worry in which position it's going to sit. As far as the Gaur, when you dart, it has to be external recumbency because the regurgitation should not be there, aspiration should not be there.
So far, the execution has been perfect. But reality is a playground where the result doesn't always match the effort.
It is not necessary that every time you dart an animal, it's going to fall down in a place which is very conducive to lift the animal. So very quick decisions needs to be made.
Everything was going well. The third animal we captured on first day in Ghana. Animal went on a bad place and he just moved into the bad position. So we just lifted the animal, went for the revival very immediately and did all the emergency protocol which has to be followed. The team was full of people who have the expertise in the emergency handlings. But at the end we lost the animal. This gave us a learning. This gave us learning terms. Because that one animal gave us so many things to go on and the life never go waste. Because losing an animal, the entire team who is involved is entire capture operation, from the stretcher to the ground team to the veterinary to the officials. Everybody gathered there. We just had a brief talk about the situation. So we moved ahead with same energy, same zeal and same enthusiasm. People are ready to go.
The team has suffered a big blow, but they must pick themselves up and use failure as fuel. The pressure is on, the tension palpable.
Yes, very good.
But the team stays calm and sees the capture through safely.
What I found to be wonderful about my team is that all the people from FD sir to our ADs to our range officers right down to our forest guards, they have been really into it. They understand the gravity of it. They understand why we are doing it. We have found inspiration in everyone. For example, there's one TPF Suraksha Shramik, Tularam. He's from on day one itself, he got injured in his shoulder. But since he was the leader of the team, he kept going on. Yesterday we asked him to take rest because he had swelling in his shoulders. He point blank he refused. He said, I have to be part of it. So he just said, I will use my other shoulder, but I'll be part of this. So this is something the commitment that you see from the team in Kana, it really inspires you. It makes you go on and on.
The Gaur gets feisty. Dr. Abhay sustains a minor injury in the melee. The team has bounced back from a heartbreak strongly, but there's a long way to go. And a big test awaits. A massive bull, much heavier than the team has handled before. It takes superhuman effort to lift and move him safely. But because superhumans don't exist, we rely on teams. And when a team works together, no giant is too big. And no bridge too far. As successful as the team has been in Kanha to facilitate a healthy gene pool in Sanjay, they must take Gaur from at least 2 different source populations. So they turn to the central Indian highlands for more. On the slopes, plains, and amid the gorges, ravines and rivulets of this peak-studded reserve lies a floral and faunal diversity that fully justifies its name. Translating to 7 folds. Folds of a plentiful earth shaped by the tireless carving of time's chisels.
I learned a kind of a lesson from Bandhavgarh because all the Gaur had gone there from one source population. We wanted to have 2 different, a little bit isolated population there, which is always good for a founder population. So that's why we selected Satpura as well.
And so, it's only appropriate that this coffer of biological wealth was to be the second donor of some of its bovine dwellers to reanimate Sanjay's cradle. The goal here is to emulate everything that's been learned in Kanha, but in a different kind of habitat.
Every capture and translocation is completely different, and Satpura is more challenging because we have a thick woodland and some of the terrain is not easy to approach also, so that makes very challenging here.
The Gaur found here are heavier too.
Availability of the floral species is very high. Throughout the year you see there are some or other trees are fruiting, and we have plenty of water, plenty of grass in this landscape. That makes these animals are generally bigger than other central Indian parks.
But the team proves equal to the gauntlet.
So in MP, we have been translocating for many years now, and a lot of them are what we call mass captures or passive captures, where we do not drug the animals. In Gaur, obviously, we have to put in a chemical capture. That's the only way we can transport them. And Gaur was one thing that we thought is necessary to keep in touch, because we need the staff, because it's a big operation. Lots of people involved, lot of training involved, lot of drills involved. So unless you keep on doing it, our apprehension was that people would kind of forget. So we did not want that. We want to keep all the trainings that we had, all the learnings that we had, we wanted to preserve going forward.
With the experience of captures in diverse situations, the team just carries on the great work from Kanha. More Gaur are captured in quick succession, all without a hitch. Satpura has lent another fold, this time to the fabric of Sanjay's future. Back in Kanha, over the next few months, the team captures several more Gaur, some big and some small. Some in bright sunshine, yet some in rank darkness, and some in pouring rain. In all, they get 50 individuals over a year from Kanha and Satpura, meeting their target. So Sanjay can have the population it needs to become again an abode of the big bovid.
Before starting transportation, we have to stabilize the animal.
It's very good. They are quite comfortable.
Journey is also of 14 to 16 hours. It is a challenge.
With the hardest part of the operation done, the animals are moved to Sanjay with the utmost care. The Gaur cope just well. And soon the vehicles are knocking at the gates of Sanjay, where a team waits for the return of the giants. It's late when the party reaches Sanjay, but a glorious full moon keeps the night young. As their hooves touch the earth, it's one small step for the Gaur and a giant leap for Sanjay.
बड़े प्रोजेक्ट का एक छोटा सा चरण पूरा हुआ पहला कदम.
The mission of bringing Gaur home may be complete, but for scientists such as Bhaskar, the project has only just begun. The newly formed herds will be continuously monitored for the next few years. Since their release last year, the Gaur have explored over 1,000 square kilometers of Sanjay's forests and have even been to the borders of Bandhavgarh, revealing excellent corridor connectivity between the two tiger reserves. In the past 12 months, 11 calves have been born, a crucial step towards the breeding success at Sanjay Tiger Reserve. For the core team, this experience has been a watershed.
Majestic animal came here again, so it was a great moment.
Always so enthralling and satisfying to be a part of conservation program like this.
We are fortunate to enough the part of this team and part of this operation.
I would say blessed, definitely, to get such an experience.
We broke the myth, the hitch about this temperature, adversities, we had overcome it. We are the best on planet Earth now, as far as the Gaur capture is concerned.
With the power of research, preparation, grit and commitment, the team has fought the odds and achieved the Herculean. Sanjay may be a new home for these Gaur, but for the species, it is a return to the roots. The new home is a true home. The mighty Gaur is back where it belongs. And this time, it's here to stay.
So thank you so much for watching this. It's been really nice to get this applaud also. Thanks to Nishant and Himani to really bring this whole in a single form. Really thank you each and every one of us, whoever is present here. And definitely the field staff, dedicating this whole film to the field staff, the Mahawards, elephant handlers, the captive elephants and the frontline staff. Thank you so much for watching in. Thanks a lot.